It is a great pleasure to be here in Oslo, nearly 30 years after I was awarded the Nobel Peace Prize. I wish to thank the Norwegian Nobel Institute and the US Embassy in Norway for arranging this lecture. Today I am here to take stock of the contributions of the so-called Green Revolution and explore the role of science and technology in the coming decades to improve the quantity, quality, and availability of food for all of the world's population. So began a speech by Dr. Norman Borlaug in the year 2000. He looms large in the world of plant breeding, not just because he personally did a lot of applied research that created new, higher-yielding varieties, particularly of wheat in Mexico in the 1950s, but also because he was an uncompromising and passionate advocate for the mixture of genetics, industrial chemistry, and farm machinery that we can call modern high-intensity agriculture. For Borlaug, this sort of agriculture boosts yields and saves lives, and that's it. And to stand against that is to stand for starving children and the destruction of the environment to facilitate low-yield agriculture. In the year 2000, genetic engineering of crops had arrived on the scene, and probably the most famous plant breeder of the 20th century had this to say as he wrapped up his speech. "I now say that the world has the technology to feed on a sustainable basis a population of 10 billion people. The more pertinent question today is whether farmers and ranchers will be permitted to use this new technology. Will the affluent nations can certainly afford to adopt ultra-low-risk positions and pay more for food produced by the so-called organic methods? The 1 billion chronically undernourished people of the lower-income food deficit nations cannot. It took some 10,000 years to expand food production to the current level of about 5 billion tons per year. By 2025, we will have to nearly double current production again. This cannot be done unless farmers across the world have access to current high-yielding crop production methods, as well as new biotechnological breakthroughs that can increase the yields, dependability, and nutritional quality of our basic food crops." Borlaug imagined a fight between the forces of science and technology and those against. And he saw genetically engineered crops as the next step in a journey of agricultural technology, and particularly crop genetics, that began a century earlier. How was it that the work of a few Mendelian geneticists around the year 1900 morphed into a global movement for agricultural technology that flowed right into the genetically engineered crops of today? Let's find out. Welcome to Modified. I'm your host, Dr. Orlando Delange, ex-plant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs and hopefully just enough of the history and the social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is Episode 8, How Science Took Over Plant Breeding. Do you eat a lot of rice? I do. And so, after decades of making rice very poorly, I invested in a rice cooker. What could be simpler? I just go to the cupboard, grab my bag of Calrose rice from the shelf, wash it, put it in the cooker, using the cup and the water markings. I close the lid, and the little bells and boops tell me when it's ready. Perfect every time, and every grain almost identical. And if I find I reach for the shelf and the rice bag is empty, I know that I can go to the store and find fresh bags waiting, sorted by variety, with each bag containing an identical product. How is it that I can have that confidence? How does a plant seed become a product, reliable, uniform, and predictable? Part of the answer lies in all the processing steps along the way that have weeded out misshapen grains, and in the packaging that turns a natural product into something that feels like it was manufactured in a factory. Part of the answer is the mechanization and standardization of growing techniques that allow for a more uniform crop. But the largest part of the answer is plant breeding, and the farming system connected with it. Genetically uniform seed, developed by specialist breeders and sold to farmers. This system, which first appeared around 2 centuries ago, with breeders existing to generate new varieties to buy, grow, and keep, and over time becoming the norm in much of the world to the extent that many farmers are legally forbidden from keeping or sharing the seed from the crops they grow. And it is, if you think about it, a rather strange idea. Why would a farmer buy seed rather than keep it to grow again? It's a waste of money, surely. The question is one we will come back to in Season 2 of this podcast. Sneak peek, Season 2 will be about the business of GMOs, And once we feel pretty confident of the science behind what a GMO crop is, we'll dive headfirst into the social, political, and economic influences and impacts of GMOs, with, of course, some more plant science and genetics thrown into the mix, because I can't help myself. Today in the US and many countries, rice farmers, as with other crops that are grown from seeds, mostly do not keep some of their harvest to save as the seeds for next year's crop. They will buy the seed from a company, or from some other distributor, as we'll learn a little bit more about later in the episode. But in many parts of the world, seed saving is still the norm, as it has been for millennia. A farmer will take care to only harvest seed from the plants at the center of the field to make sure it isn't accidentally mixed via pollination with some other farmer's field. They will take care to keep only high-quality seeds and make sure they are maintained in a cool, dry storage vessel to planting time. They may trade locally with other farmers, acquiring new varieties or selling some of their own. At each generation, they will look for the plants that are the most robust, that weather environmental shocks, and produce the largest yield. They are both preserving and also genetically improving one or more cultivars of the crop, without ever involving the scientific tools that we'll learn about through the rest of this episode. There is a key distinction between the cultivars developed through this traditional method and the cultivars that can be purchased from a plant breeder. The farmer-developed cultivars are genetically diverse. They are gathered at each generation from multiple plants, each pollinated by other plants, and even though most of that pollination will be happening within a field, sometimes pollen will float in from other fields. And then that genetic diversity will be increased whenever farmers trade seed with each other. The cultivars developed in this way are known today as land races. Cultivars of a crop that are genetically very similar but not totally uniform, and are the product of often centuries of selective breeding in a limited geographic area. There are many advantages to land races. There is enough genetic uniformity to have a pretty reliable idea of what to expect from your plants, and through generations of selection, the genetic traits of the plant are well suited to the microenvironments and growing practices of the region. But there's enough genetic diversity to make the overall crop more robust against variations in temperature, rainfall, and diseases. Within your field, you will have at least some plants that will survive most conditions. You won't lose it all. Land races are an easy way to create crop varieties that work well if your goal is to reliably produce enough food to eat and a small surplus to trade with. But they are not the good basis to develop varieties that yield enough to get a huge surplus, or to develop varieties with specific desired properties, e.g. shape, color, frost tolerance, etc., and doing so in a targeted, deliberate way. The scientific plant breeding that emerged in the early 1900s offered just that. Plants that could yield a huge bounty of almost designer products, with traits sculpted based on what farmers and consumers wanted to see from that botanical product. Plant breeding is a story of creation and innovation, always striving for something new, sometimes just a slight improvement on what exists, sometimes a dramatic deviation from what came before. And yet that process of creation is also one that has reduced diversity, taking the work of breeding crop varieties from something distributed across millions of minds and hands into the restricted domain of thousands of specialists. It is a familiar story in the history of global economics, the separation of a complex process, in this case agriculture, into a set of more siloed specialties, even separating out the growing of crop plants to sell as a final product from a specialized craft of growing fields of those same plants just to harvest seed to sell to those other growers. Seed grower, separated from farmer, separated from breeder, all relying on machinery and chemical inputs derived from their own industries, with their own specializations and divisions of labor. Plant breeding is in some ways thousands of years older than genetics, in that farmers have been breeding new plant varieties everywhere and every when that agriculture has been a practice on our planet. And even organized plant breeding with dedicated specialists is many centuries older. Medieval imperial China had a state-run system of science and technology support for farmers, including in provision of seed, in a system that sounds quite reminiscent of the government-funded systems that have become common to most countries since the mid-20th century. Scientific plant breeding was all the rage in 18th century Europe, long before anyone knew what mechanisms were responsible for the observable ability of plants to hybridize and produce offspring with new trait combinations. The appearance of the first commercial enterprise of breeding and selling seed on the promise of improved performance for growers is hard to pin down, but an early notable example is the Vilmorin Seed Company, founded in France in 1840, and it's still around today. No one needed Mendel and the geneticists to tell them that plants could be bred to produce new varieties, and that people with time, careful observation, and good business sense could turn that into a profitable enterprise, well suited to the 19th century and its growing industrialization of agriculture, along with the industrialization of everything else. The first steam-powered tractors appeared in the 1860s. New machines, new seeds, new models of labor and wealth accumulation. Plant breeding carried out in parallel to genetics, even after William Bateson and the other geneticists of the early 20th century had managed to push genetics into the scientific mainstream. One of the most famous plant breeders ever is Luther Burbank, child of Massachusetts who became hugely famous and quite rich in California in the 19-teens. His many enduring contributions to American agriculture and horticulture include the russet potato, the Shasta daisy, and the plumcot. He also selected and promoted existing varieties sourced from all around the world. These include the Himalayan blackberry, which is now an invasive species in the Pacific Northwest, thanks to Luther Burbank. It is a major pest ecologically, but it does have great berries. Luther Burbank was an early 20th century celebrity, seen as the equal of Henry Ford and Thomas Edison. A really strange guy, as seems to be a requirement for celebrity inventors, and I highly encourage reading The Garden of Invention, a biography of Burbank by Jane Smith. He achieved his success in plant breeding despite rejecting genetics completely. He had his own theories about what was happening inside his plants, which he did try to promote at scientific lectures at universities, but which all fell apart quite quickly under scrutiny. Still, no one could doubt his success in breeding and selling new varieties, and he did it all without a hint of Mendelianism. So plant breeding didn't and doesn't need to be connected to genetics. And yet, they became, plant breeding and genetics, more and more intertwined as the 20th century wore on. Everywhere around the world that plant breeding is done at scale, whether by companies or government agencies, you will find the concepts and tools and the knowledge systems of genetics. The strange consequence of the integration of genetics and plant breeding is an entanglement between the various institutions and social constructs that make up professional science with those of agriculture, and of both of these with state governments. I won't take us through a detailed chronology of that integration. I'm not a historian, so I don't think I'd trust myself, and it would take many, many hours, and this is already going to be a pretty long episode as it is. And after all, there is episode 8 of a podcast about GMOs, and we've barely talked about GMOs directly, so I think we should keep things moving at a decent clip. So instead, I'll just look at 3 vignettes from the 20th century that provide examples of how scientists, particularly geneticists, pushed their way into plant breeding to get to the point today where genetics and related scientific disciplines are fully embedded in plant breeding. We will look at Mendelian breeding in the earlier 20th century, mutation breeding in the mid-century, and the dawn of marker-assisted breeding towards the end of the 20th century. Vignette 1. Mendelian breeding in the earlier 20th century. Working roughly chronologically, Mendelianism muscled its way into plant breeding from the very start of the 20th century. As we explored in a previous episode, British Mendelian William Bateson was a huge booster when it came to the synergy between his favoured science and plant breeding, and he surrounded himself in his professional connections with like-minded people like Roland Biffin and Reginald Punnett. Yes, the Punnett who came up with a system of tracking Mendelian inheritance using squares, still used in classrooms the world over. That is, the Punnett square. In the early decades, what the Mendelians gave to plant breeders, more than anything, was not a precise new set of techniques that required laboratory equipment and advanced math. Instead, it was more about a different strategy. Traditionally, plant breeding has been based on mutation and selection. Mutations meaning that farmers and pre-Mendelian plant breeders would look for unusual individuals with desirable characteristics and use those as a new breeding stock. And selection refers to the guiding hand of the professional breeder or farmer over generations of the crop, always looking for the biggest or the hardiest, selecting those individuals to be the parents for the next generation. That approach was given a huge boost by the runaway success of Darwin's theory of evolution by natural selection. Darwin's theory was supported by the long-standing practice of selection by plant and animal breeders, and his twist was explaining how the same process, without any conscious guidance, could actually happen in nature to change species over time. But this approach to breeding, mutation and selection, it does work well. It has worked well for millennia. And mutation in particular actually got a big power-up in the 20th century with the help of nuclear power, as I'll explain shortly. However, relying on mutations that happen by chance is not a great business strategy, and selection itself is quite slow, and it's always hard to predict the results. He was wrong, but William Bateson actually didn't believe in selection. He was highly suspicious about Darwin's theory of evolution via natural selection, because to him, it didn't mesh well with Mendelian genetics. One of the major projects of the 20th century was unpicking how both can in fact be correct and are correct. But for Bateson, traits don't just gradually shift over time. Instead, traits are the product of discrete entities called genes, which can have different versions called alleles, and that variation is only the product of different allele combinations in any individual. The Mendelian approach to breeding rejected selection in favor of careful hybridization. Not that no one had ever thought to hybridize plants before, both varieties within the same species and even between different species. As an aside, the whole idea of species as things that can't successfully interbreed is a very animal-centric idea. Plant species hybridize all the time, particularly in the hands of an ambitious plant breeder. While plant hybridization in the past was more like a random roll of the dice, the Mendelian approach was to consider what are the traits you would like to see in your new plant variety. Find those in existing varieties, whether domesticated or wild, and then through a series of controlled crosses, combine all of them into the same lineage. You don't necessarily need anything more than patience and faith for this approach to work, but those are not attributes that should be underestimated. Think about it. What the Mendelians believed correctly was that inside every plant are untold genes with different possible alleles, and that for each gene a given plant can have multiple alleles, and that it is common for some alleles to be dominant and others recessive, such that you do not see the effect of the recessive allele if they are paired with a dominant one. That means that the traits you might be looking for could be hiding within a given generation of plants in the form of recessive alleles. It would be very easy under such circumstances to lose interest in trying to carefully combine traits from multiple offspring. It requires a lot of trust that your traits are in there, hidden, waiting to re-emerge with the right allele combination, rather than the more common sense understanding that the traits simply all blend together in every generation in hard to predict ways. One of the particular challenges the early Mendelians like Bateson and Punnett set their sights on was rogues. These are plants that seem to grow very differently to the others around them. There were many theories about what they were, with the prevailing idea, at least in the late 19th century, being that they are reversions to some ancestral, remembered type. There was no room for such a mechanism as this within Mendelianism, and so Bateson made it a mission to show that actually, in almost every case, what seemed to be a rogue was the result of some seed from the wrong variety getting mixed into a seed bag, or pollen from a neighboring field wafting over in the case of farmers keeping and sowing their own seed. The Mendelians urged plant breeders to stand their ground and trust in the idea of hybridization as the basis of plant breeding. And which plant breeder would want to say they are going to reject what, at the time, was very cutting-edge, exciting science? They didn't want to appear behind the times. The Mendelians did also offer techniques to support their new approach. For example, explaining to plant breeders how different types of crosses can be used to reveal what allele combinations are present within each breeding generation of plants. And over time, these techniques became integrated into the norms of plant breeding. Patient selection over many generations and screening for random mutations got replaced by a methodical use of crosses between existing varieties to create new trait combinations. Vignette 2. Mutation breeding in the mid-century. So after talking all about how genetics encouraged breeders to give up relying on random mutations, I'll now talk about how it did the opposite just a few decades later. So I was talking about rice earlier in the episode and how uniform every grain in a bag of rice is. And it happens that Calrose is the rice I buy most often. So let's use Calrose as our example for mutation breeding. And it just so happens to connect back to what we learned about gibberellins in episode 4. I have to say I was a little surprised as I researched this episode to find that California is one of only 3 states in the US with a major rice agriculture industry. And California growers overwhelmingly produce Calrose rice, a medium-grained, mildly flavored rice that was first developed in 1948 at the Rice Experiment Station in Biggs, California. The Rice Experiment Station, or RES, continues to provide California growers with updated and improved versions of Calrose rice. The RES is just one example of a very common kind of institution, the Cooperative Plant Breeding Institute. It was created at the very start of the 20th century by farmers in California who were interested in growing rice but needed a reliable supply of seeds that would grow well in California, that needs a mixture of plant breeding expertise and infrastructure to produce and distribute seed. The farmers pooled money via subscriptions, and the federal government chipped in a bit, and it's more or less continued that way since. Essentially, a farmer cooperative with government support. And that is a pretty common system in different regions and for different crops. The world of agriculture is not just a bunch of private businesses out there being ruggedly individual. There is a lot of cooperativity. Anyway, that's not the main thing I wanted to talk about. Instead, I wanted to talk about Calrose 76, just as one of many possible examples of a crop variety that was created using mutation breeding. There are many ways to do mutation breeding, but at its heart, what you're doing is mass mutating a bunch of seeds and then growing them and looking for useful new traits or improved versions of traits, e.g. bigger grains, better heat tolerance. So how do you mutate the seeds? Well, the most popular way to do this is using gamma radiation. Gamma rays are high-energy electromagnetic waves. So visible light, radio waves, and X-rays are all electromagnetic waves of different types. Gamma rays are tiny, high-energy bombardments, and they are small enough to be able to directly interact with atoms, including the atoms that make up DNA. And in the process, they can break DNA and trigger an error-prone repair pathway that changes the sequence of DNA wherever the gamma rays did their damage. To use genetics terminology, radiation can be used to create new alleles. But these new alleles are random. You don't know which genes will be hit and quite what mutation you'll get. So with mutation breeding, it's a numbers game. You need to mutate a lot of seeds and screen the resultant mutant plants in large numbers to find any useful alleles. Radiation, like Mendelian genetics, was a field of study that really was born in the start of the 20th century, and it took a while to work out what radiation is, what it can do to different materials, including DNA, and also how to produce it in a controlled way. Because of that, radiation breeding only really got underway in the 1960s. And it helps to correct the issue with plant breeding as pushed by the Mendelians. They opposed the idea of just waiting around for random mutations, and instead encouraged seeing plant varieties of sets of traits that could be combined in predictable ways using genetics. But that does limit you to only the traits that exist within available varieties as well as wild relatives. But sometimes the trait you want just isn't there. And so mutation breeding allows you to basically inject new genetic diversity into the available stocks, and within that, hopefully some traits that you can use and introduce into your breeding programs. The mutation with gamma rays is just a way to create more variety to feed into the crosses you're doing, rather than an alternative way to create crop varieties. Remember that, because the same thing is true with GMOs. Mutation breeding can also be used to introduce a specific genetic variation into an existing crop variety, and that gets us back to the RES in Biggs, California. It's pretty random, but I picked it to use as an example of mutation breeding. Specifically, it was the site of the creation of a new variety of Calrose in 1976 using mutation breeding, the imaginatively named Calrose 76, created by J. Neil Rutger. In the show notes, I'll link a video oral history with Dr. Rutger from the UC Davis Aggie Video Archive, because there's a lot of interesting stuff in there, among a lot of very dry chat about plant breeding careers in the 70s. By the 1970s, a lot of work had been done in Asian rice varieties to identify shorter, higher-yielding varieties. If you remember back a few episodes, you might recall the plant hormone gibberellin. These shorter, higher-yielding rice varieties have a mutation in a gene that we now know encodes an enzyme involved in synthesizing gibberellins inside plants. So mutations in this gene reduce gibberellin production. Gibberellins promote cell expansion, and so these plants have shorter stems, and the energy saved from investing in stems is rerouted to invest more in grains. So you get shorter plants with bigger grains, and they're also less likely to bend and fall over, so you get a better harvest from them. The problem is that these mutants were available in rice varieties, but varieties that had a bunch of other traits encoded by alleles that made them great for growing in different East Asian climates and soils, and with properties that were attractive to consumers in those regions. Calrose was an existing brand, essentially, and had properties that suited California growers and consumers. It could take a long time to essentially isolate and introduce the semi-dwarf trait, as it's called, those short plants, into the Calrose rice. So mutation breeding was used as a shortcut. Just take the existing Calrose rice, well, a bunch of rice grains, irradiate them with gamma rays, and then throw them into a field and look for shorter rice plants. There are about 60,000 genes in the genome of a typical rice variety. So if you basically know that what you want is a mutation in one of those genes, but you can't control which one you get the mutation in, then you have to mutate, grow, and screen at least 60,000 grains. It's a lot, but it's actually probably faster and easier than doing the multiple rounds of crosses you need to introduce a single specific allele from a genetically distant variety. And that's just what Jay Rutker did. Well, he led a whole team of people doing it. These days, with the help of drones and AI scanning of fields, it's actually much easier than it would be, and it could be one person's work. Calrose 76 was a hit, though you won't find it grown in fields today. Instead, the semi-dwarf trait from Calrose 76 was bred into other Calrose varieties, so it is still part of the ancestry of the Calrose rice you can buy from a store today. A fun twist in this story is the role of the International Atomic Energy Agency. The IAEA was founded in 1937. Now, I'm not an expert on this, so I could be wrong, but it does seem like it's not a coincidence that mutation breeding started taking off just a few years later. It's definitely not a total coincidence, it's just a question of how pivotal a role the IAEA played. They were indeed actively promoting the use of radiation in plant breeding from their earliest days, in line with their charter as a UN agency dedicated to promoting the use of nuclear energy for non-military purposes. Basically, the IAEA is all about opposing nuclear war and promoting nuclear peace. And the IAEA directly promoted Dr. Rutker's work at the RES, providing funding to support bringing over a scientist, Dr. Hu, from Taiwan to support the project. And the IAEA continues to this day to support mutation breeding. Mutation breeding has fallen out of favor in many parts of the world, in no small part because genetic engineering now offers better ways to do similar things, by introducing specific new genetic variations rather than random ones. But it is apparently still a popular method in less well-resourced parts of the world, because it is relatively inexpensive and, well, it works. Okay, let's move on to our final vignette, marker-assisted breeding in the later 20th century. This drops us off nicely to our next episode, which will focus on the genetic tools developed in that period that enabled the creation of genetically modified crops. There's something I've hinted at a few times but not really addressed so far as we're talking about genetics and plant breeding. Most traits are not controlled by a single gene. Yes, there are examples, like in the case of gibberellin signaling genes, where a mutation in a single gene can have a major effect on a trait like plant height. And there are gene-for-gene interactions that mean that individual resistance genes can be introduced into a plant variety and that will give it a significant resistance boost against a target pathogen. But in so many cases, there just aren't individual genes that can be mutated to give a desired trait. Examples of traits like this include most traits that define growth tolerances to environmental conditions, drought tolerance, cold tolerance, acid soil tolerance, as well as things like fruit or grain size. And these are very important traits for plant breeders. These are so-called quantitative traits, as opposed to qualitative traits. They change by small quantities along a spectrum, rather than seeing big qualitative differences from a single allele change. So while the Mendelians were very loud in public in the first decades of the 20th century about having unlocked the key to a plant breeding, they really didn't have a way to help breeders who were working with quantitative traits. It took a few more decades for statisticians such as Ronald Fisher to come up with mathematical tools to track quantitative traits in predictable ways, and then plant breeders had to work out what that would mean in practice. Basically, what you could do is screen the cold tolerance of 100 different existing varieties of your crop, score them all for cold tolerance, and now you can begin the long, slow process of crossing them in different combinations to combine as many of the beneficial alleles as possible. The problem is you can't see these alleles. You suspect that somewhere in the plant are a bunch of genes with functions related to cold, and that different alleles improve or release or reduce cold tolerance. But you don't actually know where these alleles are. You can't see or track them. You can just use statistical models to make some educated guesses about the number of genes you're probably working with and things like that. It's also hard to do this quantitative trait breeding work because it's really not obvious just from looking at the plants which ones have the alleles you want. Remember, you're dealing with alleles that give subtle shifts in quantitative traits. A new tool entered the toolkit in the 1990s, marker-assisted breeding, and relies on a technique that I think is worth spending a couple minutes with because it's so deeply embedded in modern life science: PCR. Another Nobel Prize-winning invention, and for this the Nobel Prize went to Kary Mullis, California-based chemist who developed the technique while working as a research scientist at Cetus Corporation, one of the world's first biotechnology companies. In 1983, he came up with the idea of just distilling down a core biological function, replicating DNA into something that can be done outside of a cell in a test tube. And it needs just one protein, DNA polymerase. In a tube, you can combine some DNA template material that you know somewhere in it has a section you're interested in. And you add some short DNA pieces, each just 10 to 20 DNA bases long, one with a specific sequence that matches the beginning of your target region, and the other that matches the end. Mix together the DNA polymerase with the template and those primers and a bunch of the raw A's, T's, G's, and C's, the DNA bases, into a test tube, or actually a tiny, tiny little tube called a PCR tube. And the enzyme, DNA polymerase, will get to town making copies of the region defined by the short start and end pieces, the primers. The only help along the way is that you need to repeatedly boil the whole mixture to pull apart the strands of DNA double helix so that the DNA polymerase can access single strands to replicate. In a cell, there are other enzymes to do this work without needing to boil anything. Outside of a cell, we use machines called thermocyclers that cycle the temperature up and down in cycles. The amount of DNA copies in the tube doubles at each cycle. So if you start with just 2 copies, you will have 8 after 2 cycles, and an astonishing 1 billion copies after 30 cycles. That's enough that you can run that DNA through a gel with tiny pores that separate DNA molecules by size, and by adding a stain that makes DNA visible, you can actually see the different fragments of DNA and what size they are. And all of this takes a few hours, and you can easily scale that up to do tens or hundreds of these PCR reactions in parallel. This I know because I do them all the time. PCR has got to be one of the most common practices in laboratories all around the world. You likely received a PCR test for COVID at some point. That was using short DNA pieces, primers, that match sequences in the COVID genome. You need a template piece of DNA to kick the whole PCR reaction off. So that means basically you run the PCR on the patient sample, then you look at the results. If DNA indeed got copied, then there was some COVID template DNA in the sample to begin with, aka the patient has COVID. If you don't get any result, no DNA on the gel, then there was no COVID DNA in there to start with, aka a negative result. And yes, I know that the virus is SARS-CoV-2 and not COVID, it's just for brevity. What PCR does is provide myriad variants on the basic theme of making invisible genetic differences quickly and easily visible as different patterns in the results of a PCR reaction. PCR can be used to look for just one thing, like a PCR test for COVID, but it can also be used to take a whole genome and basically sample DNA sections from across the genome and produce a visible pattern in the PCR results. And that even if you don't know how this pattern relates mechanistically to crop traits of interests, it gives you DNA features that you can at least correlate to traits in crops. Remember that till this point, roughly the 1980s, plant breeders have been thinking about crops as collections of genes that they know are somewhere in the genomes. Since the 1950s, the physical nature of genes was increasingly well understood, but there was not a quick and easy way to connect the traits of a crop to the physical properties of its own unique genome. PCR allowed that. You can imagine it like being able to get a fingerprint of the unique DNA features of a plant. And what's more exciting, you can do it with just a tiny, tiny amount of starting material, easily plucked off of a growing seedling, or from a seed pulled from a sample of genetically uniform seed stock. Now, you do not have to wait a full generation to check the phenotype, and to use that phenotype to indirectly infer the underlying DNA features. You can go straight to the source, saving time. Conceptually, the work shifts to being all about trying to combine genetic lineages in a way that gives you a new target DNA fingerprint that contains as many DNA features as possible that you know tend to correlate with your trait of interest. No one DNA feature within the overall pattern is the gene for X, the gene for cold tolerance. But you know that there are lots of DNA features that correlate with cold tolerance, and now you can easily check how well you are combining all of these into your new crop variety. And yes, of course, in this process, you do have to grow some plants to maturity so that you can cross them with other varieties and produce this new variation. And in that process, you will be checking for your trait of interest, such as cold tolerance. But a lot of that work can now be done very quickly inside a lab, instead of over months working in a field. Hopefully this makes some sense. Sorry that I'm still being quite vague. To go into the details more literally and less metaphorically would require several episodes. And I figure it's better that we keep moving towards GMOs. And I will spend several episodes diving into more detail of exactly how GMOs are made in the coming episodes. But if you want to hear more about other kinds of plant breeding methods and how they work deeper than what we've covered so far, do let me know and I can come back and add more detail in a future episode. But for now, let's finish up. Arriving in our minds in the 1990s, genetics has completely embedded itself into plant breeding. And plant breeding is just one aspect of an industrial approach to agriculture, which has long become dominant in the US and much, if not most, of the world. Norman Borlaug may have had his complaints about what he saw as a growing opposition to industrial agriculture, but when he died in 2009, I think he would have felt broadly satisfied that the approaches he dedicated his life to had won the day. By the end of the 20th century, crop plants weren't things that farmers in most of the world grew from the seeds they themselves produced, based on locally developed and maintained knowledge. That had been outsourced. Private plant breeders, as well as working cooperative and government-run agricultural centers, provide farmers with a range of products. Seed stocks carefully bred to maximize yields based on different conditions and different types of farm. A whole marketplace for the farmer to choose from. Carefully modified genetic stock developed by experts in government and private research facilities. So when some researchers in San Francisco in the 1970s showed that genes are not just things you can describe and even see, but things that you can move from one organism to another, even between organisms separated by billions of years of evolution, well, why wouldn't plant breeders immediately want to integrate that amazing potential into their suite of scientific tools? But we'll pick up that thread on our next episode. For now, I want to particularly acknowledge Beres Charnley's 2011 dissertation from their PhD at the University of Leeds, Agricultural Science, Plant Breeding and the Emergence of a Mendelian System in Britain, 1880 to 1930. And I want to acknowledge nobelprize.org, where I got the full text of Norman Borlaug's 2000 speech. You can find a lot of speeches from prominent scientists in their archives. This has been Modified by me, Orlando Delange. If you're enjoying the podcast, please do subscribe and tell a friend. The number of listeners has been inching up, but if you know someone who you think might be interested, please do let them know. And if you want to get in touch with me, please do. You can email me at orlando@modifiedpod.com, and you can find my Blue Sky and Instagram handles to message me that way over the website for the podcast at modifiedpod.com. And with that, I hope you'll listen again soon. Bye!